Hi everyone, this is Mr. Hall and this is a quick video about the small intestines and absorption. By the end of today's video we'll go through the following things. There's quite a few things to go through. We're going to describe the role of the small intestine. We're going to explain why starch can't pass from the small intestine into the bloodstream. We'll describe and explain the adaptations of the villi for absorption, and we'll also describe the process of active transport in the small intestine as well. So first off, let's start about where the small intestine is. Now, in previous videos I've talked about lots of the different organs in the small intestine, and previously I've talked about how food sort of starts in the mouth and then it goes all the way down the esophagus into the stomach, which I know looks a bit confusing because the liver covers it, but the stomach is basically this organ over here. So food lands into the stomach, it starts digestion, and then after that it then moves into the small intestine. And the small intestine is this sort of very wiggly long organ here. Okay, so food makes its way through there to digest further. Now, in this part of the digestive system, it's also the place where the nutrients that have been digested pass from the digestive system into the bloodstream, and then from the bloodstream it can get carried to all the cells in your body for use in respiration, amongst other things. Now, some digestion also happens here, in particular lipids or lipids, otherwise known as fats. They're broken down by the enzyme lipase. Now, what I've shown you here, it's basically a stretch of the small intestine. That's the pink part here. So I'll change the ink color just to make it a bit clearer to see. So this whole sort of tube over here, this is your small intestine, okay? And then what you've got are two different types of things. These small yellow circles, they represent the smaller molecules, for example, of glucose. And then the larger ones here, this is typical of what you'd see, for example, like starch. And as you may recall, starch is a very long carbohydrate chain. And then when it gets broken down by carbohydrates, it turns into glucose. And then on the outside, this red part here, that, if I just change the color again, this represents your bloodstream. Again, the bloodstream sort of surrounds it on all sides. So now these nutrients, they are absorbed from the small intestine into the bloodstream via diffusion. So these food molecules you can see here, they'll eventually move from where they currently are, and then they'll eventually sort of move out into the bloodstream like so. Now, the concentrations of the nutrients you'll notice is greater in the bloodstream than in the small intestine. So you can see there's lots and lots of glucose in the small intestine here, not so much in the small intestine. So it can't move normally, it can't move by normal diffusion going from an area of high concentration to low. It has to go against a concentration gradient. So as a result, it has to move via active transport, which requires energy, and the energy can be provided from glucose, which is already in the bloodstream. Now, these large insoluble molecules, such as starch, they cannot be absorbed. Now, there's a couple of reasons why that can't go there, so that can't happen. One of the reasons that it's simply too large, so it can't fit into the very small gaps that you've got in the walls of the small intestine. The second reason is because it's pretty much insoluble, and it has to be soluble in order to pass through. So there's a couple of reasons why it can't move through. And the ones that can move through will move through in that following way. Now, a popular way that you might be asked about this particular type of digestion is through an experiment. And this experiment is used quite a lot, or demo, I should say, is used quite a lot to show this process. So what you'll get is this kind of rubber tubing. We call it a kind of plasticky tubing. We call it a visquing tubing, okay? And inside this visquing tubing, we put in starch. Now, if you remember, starch is this really, really long molecule, okay? And it's a large carbohydrate, and it's not very soluble, to be honest. So although we call it a solution, it's kind of, you can see the bits of starch sort of floating in solution as a suspension, if you like, and it can't go through, so that can't happen. And then on the outside you've got iodine. Now, iodine is kind of orange, okay? So what you've got at the very start here is an orange solution, and then in here you've got sort of like this cloudy solution at the start. And then what you can then do is, whilst you get that whole thing set up, you can then put an enzyme inside here. In this case, it would be carbohydrates or amylase, and that will start breaking this down into smaller pieces. And then what you can then do is you can see the change in the iodine solution color, and you can also add Benedict solution to see how the color changes of that too, to see how the digestion changes as a result. So a couple of things happen here. So first off, if we have this at the beginning and this is sort of later after, let's say, about 15 minutes. Now what you can do is you can also put iodine inside there if you really wanted. Now, if you think about it, at the beginning, inside the gut, if I put iodine inside there, it would be blue-black. All right, and the reason for that is because it's full of starch, so it's going to turn blue-black in the presence of iodine, as you may have learned from food tests before. And then the water around it, the iodine there will be orange because there's no starch inside here. Now, after about 15 minutes, this starch, which is kind of this long molecule here, will then break up into smaller pieces. So if you look at this after 15 minutes, you'll have small bits of glucose, which will then be able to move through. Okay, you might get 1 or 2 molecules of starch remaining, but not many. But the key thing is most of the starch is broken down. Now, because most of it's broken down, and this blue-black color will actually start to disappear, and you'll be left with orange. And then the water around the gut, because no starch has managed to move through, it will also remain orange. Okay, and then just to fill out this third column, does the liquid contain starch? Well, at the beginning it's a yes, but the other times it's no because it gets broken down. Now, what you can also do is you can add Benedict's reagent. Now, Benedict's reagent is a really good test to see if something contains sugar, such as glucose. Okay, so it's basically like a sugar test. And if it's blue, there's no sugar. If it's brick red, there is sugar. Now, at the beginning, you can see that all I've got inside the tube is loads of starch. There's no glucose at the beginning because nothing's been digested. So if I was to put Benedict's solution inside here and here, it would simply remain blue. So that would be blue, and that would be blue. Now, over time, as I showed you here, that starch is getting broken down into glucose, and then the glucose is going to start to diffuse and move through the viscine tubing because the viscine tubing is partially permeable. So you're going to have glucose here, and you're going to have glucose here. Now, because of that, that blue color of the Benedict's reagent is now going to start to turn brick red. So it's going to kind of all look quite red color. So that will look red and that will look red. And the reason for that is because you now have glucose, whereas at the beginning you didn't. This experiment comes up a fair bit, so it's good to be familiar with that demo that might come up. Now I want to kind of just return to the absorption of nutrients in the small intestine. Now, the small intestine is an exchange surface that is specifically adapted for its job in a similar way to other exchange surfaces are, like the alveoli or root hair cells or fish gills, stuff like that. And the thing which my diagram doesn't show particularly well is the lining of my small intestine. Now, at the moment, it looks relatively smooth, but actually what you've got, if I just zoom in on it, is something far more sophisticated. What you get is this small little kind of tube things called villi, and they are on the surface inside your small intestine. Now, what we're going to do is we're actually going to zoom in on one of these villi up close to see what it looks like. And what it looks like is basically like this. So this is one of those small tubes, and you can see even then that small tube has mini tubes in it too. Now, those mini tubes are microvilli, okay? Those tiny things. So the whole thing is a villi, but the smaller ones are microvilli. And these increase the surface area of the small intestine up to a factor of about 500. So it massively increases surface area. And then what you'll also notice, you'll see this red line here, and that represents the blood vessel. And you'll notice that it's really, really close to the surface of the villi. Now, all of these things help to absorb nutrients into the bloodstream as efficiently as possible, okay? And I like to use the acronym TLC in order to help remember these key things. Now, the T stands for thin membrane. And what it is basically, it means the surface, the microvilli, it's one cell thick. So that diffusion path is really short. So it makes that absorption as quick as possible. The L is large surface area. We've talked about that already. That maximizes direct diffusion, makes it happen really quickly. And then the C is for the capillaries, which are the tiny blood vessels that are on just under the surface of the villi. And they basically have two roles. The first one is they're close to the surface, so the diffusion path is short. And that basically means that if I'm a nutrient here, I don't have to travel very far in order to get to the blood. And the shorter that distance, the more efficient your absorption. And then secondly, you've got this blood supply which is constantly moving. I can't really show it on my diagram, but it's constantly moving. And as it's constantly moving, it's moving away the nutrients that have been absorbed and taking it to the rest of the body. And by doing that, it's trying to maintain the steeper concentration gradient as possible. And that's quite important because of the active transport that you've got going on here. And that's it. So I hope that video has been helpful, and best of luck with your studies on the small intestine.